Yes, we live now. Welcome to Let's Learn Bio by NCA. This is Nirupadesh Amdiyal. In this session, we are going to discuss biotechnology and its applications. Within that, the biotechnological application in agriculture. Within that, the first question: what is an explant? Explant, it is the part of a plant used in tissue culture is called explant. Second option. Next question: the inherent capacity of a plant cell to regenerate whole plant from any cell or explant, it is called, that is a totipotency. Next, third question: green revolution resulted in improvement in crop yield and production of food grains due to green revolution, tripling food production, that is mainly due to the use of agrochemicals, better crop management practice or techniques, introduction of high yielding varieties with improved germplasm. All of these. Next question: the isolated protoplasts from two different varieties of plants can be fused to get hybrid protoplasts, which can be further grown to form a new plant. This process is called somatic hybridization. The example for this, the tomato. Tomato, it is produced by the fusion of two protoplasts of one tomato and tomato plants together produced somatic hybrid called tomato. Micropropagation, it is the production of thousands of plantlets. Somaclonal variation introduced during tissue culture, somaclonal variation. Meristem culture, it is also useful to recover healthy plant from diseased plant. Next question: given below are two statements. Statement 1: the green revolution succeeded in tripling the food supply, but yet it was not enough to feed the growing human population. This is correct statement. Second statement: tissue culture was developed because traditional breeding techniques failed to keep pace with the demand and to provide a sufficiently fast and efficient system for crop improvement. Here, the both first and second statements are correct. For that, fourth option is correct. Next question: which of the following are the advantages of tissue culture techniques? New plantlets can be grown in a short period of time. New plantlets and plants are more likely to be free of virus and disease. Yes, correct. The process can be performed throughout the year. Yes, definitely. No season for the tissue culture. These help us to produce more copies of the same plant with the desired character. Yes, definitely. So the fourth, A, B, C, D, all options are correct. That is a fourth option. Next question: each plant produced by tissue culture will be genetically identical to the original plant from which they were grown. That is, that those plants are referred as soma clones. Callus, it is a undifferentiated mass of a cell called callus. Explant, it is any plant part taken out from the plant and it is used during a tissue culture explant. It's already discussed. Hybrid, the individual expressing a mixture of character called hybrid. Seventh, for this, third option is correct. Next, when a protoplast of a tomato is fused with the potato and then they are grown to form a new hybrid plants, combining tomato and potato characteristic, it is called pomato. Just already told, pomato is the best example for somatic hybrid. Next question: isolated protoplasts from two different varieties of plants, each having a desirable character, can be fused to get hybrid protoplasts, which can be further grown to form a new plant. These hybrids are called somatic hybrids. During a somatic hybridization, the products are called somatic hybrids. Soma clones are during a tissue culture, the thousands of plants were obtained. Obtained plants are resembles identical to the original plant. These are called soma clones. Embryo culture, by the cultivation of embryo, all plant could be generated, that is embryo culture. Callus, it is undifferentiated mass of cell. For this, second option is correct. Next question, transposons are referred as mobile genetic elements or jumping genes. Second option. Yes, the next question, in Bacillus thuringiensis, the bacteria itself is not killed by the toxic protein crystals because it is present in an inactive state in bacteria, that is called protoxin form. Third option. Next, the RNA interference technique is used successfully to control the nematode. A nematode called Melidogyne incognita. Fourth option. Next, the toxin of Bacillus thuringiensis is activated by the Bt toxin. It is activated by the alkaline pH of insect gut. Third option. Next, which one of the following statements are correct regarding the process of RNA interference? RNAi has been used to prevent nematode infestation of tobacco plants. Correct. RNAi takes place in all eukaryotic organisms as a method of cellular defense. Correct. The method involves silencing of specific mRNA using complementary double-stranded DNA. This is wrong. Double-stranded RNA molecule used to prevent translation of mRNA. Next, using retrovirus vector, nematode-specific genes were introduced into host plant. Agrobacterium is used as a vector to introduce nematode-specific gene into the host plant. This is again wrong. The second, third and fourth are wrong. First and second are the correct. For this, first option is correct. Which of the following is incorrect with respect to Bt toxin? Bt toxin does not kill bacteria themselves because toxin proteins occur in inactive form called protoxin. Correct. Second, toxin can kill certain insects such as dipterans, coleopterans, lepidopterans. Correct. Bt toxin is commonly used to produce herbicide-resistant GM crops. This is wrong. When an insect ingests an inactive toxin, it is converted into an active form of toxin due to the alkaline pH of gut that solubilizes protein crystals. Correct. Incorrect statement. Third one is correct. Next, what does a protoxin refers to? Protoxin, it is an inactive form of toxin, protoxin. Third option. Next, which of the following is an incorrect statement regarding genetic modification of crops? It has reduced reliance on chemical pesticides. Correct. It has reduced post-harvest loss is also correct. It has reduced efficiency of mineral usage by plants. This is wrong. It increases the efficiency of mineral usage by plants. Next, it has enhanced nutritional value of a food. The third one itself is incorrect. Next question, application of biotechnology includes. Biotechnology application includes industrial scale production of biopharmaceuticals, biologicals using genetically modified microbes, fungi, plants and animals. Correct. Therapeutics, diagnostic, bioremediation. Correct. Genetically modified crops for agriculture, processed food, waste treatment and energy production is also correct. So all of the above is right answer. Next, using agrobacterium vector dash specific genes are introduced into the host plant. Nematode specific genes here. Next, RNA interference takes place in all eukaryotic organisms as a method of dash, that is cellular defense. The genes dash and dash control cotton bollworm. Usually cry1AC, cry2AB control cotton bollworm. Dash has been introduced in Bt corn to protect the same from corn borer. Cry1AB is used to control corn borer. So for this option, third is correct. Next, golden rice is a promising transgenic crop. When released for cultivation, it will help in alleviation of vitamin A deficiency. It is a vitamin A enriched rice, golden rice. It helps to alleviate vitamin A deficiency. First option. Yes. Okay. The next, biotechnology applications in medicine. Which of the following is an example of gene therapy? The introduction of a gene for adenosine deaminase in persons suffering with severe combined immunodeficiency (SID). This is a right one. Next question. What happens to the C peptide of human insulin? C peptide, it is removed during maturation of proinsulin. In human, insulin is produced as an inactive prohormone, proinsulin. During a maturation, C peptide is removed, then it becomes active insulin. Active insulin consisting peptide A and peptide B. C peptide is removed. Next, third question. First clinical gene therapy was given in dash to a dash and dash year old girl with dash deficiency. Fill in the blank with the correct option. In the year of 1990, 4-year-old girl suffering with adenosine deaminase deficiency. Second option. Next, fourth one. Which of the following is a permanent cure for ADA deficiency? Bone marrow transplantation, it is a temporary. Injecting functional ADA into patients, again temporary. Introducing ADA genes isolated from marrow cells into cells at early embryonic stage, this is a permanent. Third option. Introducing ADA gene into lymphocytes which are subsequently returned into the patient, it is also not a permanent. Next, genetically engineered insulin was prepared by an American company called Eli Lilly in the year of 1983. Next, which of the following is an incorrect statement? The management of adult onset diabetes is possible by taking insulin at regular interval. Correct. Insulin used for diabetes was earlier extracted from pancreas of slaughtered cattle and pigs. Correct. In humans, insulin is synthesized as a prohormone. Correct. A mature insulin has a 3 peptides A, B, C. This is wrong. Mature insulin consisting only chain A and B. C chain is absent in mature insulin. What is alpha antitrypsin? Alpha antitrypsin, it is a drug used to treat emphysema. Second option. Next, pathophysiology can be defined as pathophysiology is the study of altered physiology of host after infection of a pathogen. This is pathophysiology. Second option. A single stranded DNA or RNA tagged with a radioactive molecule is allowed to hybridize to its complementary DNA in a clone of cells, followed by detection using autoradiography. Next, PCR is also early detection method. In this method, nucleic acid of pathogen is amplified. ELISA, it is based on antigen antibody interaction. It is also early detective method. Electrophoresis, it is a method of separation of DNA fragments or molecules based on their size. Third option is correct. Next, statement 1. Recombinant DNA technological process have made immense impact in the area of healthcare by enabling mass production of safe and more effective therapeutic drugs. Correct. Recombinant therapeutics do not induce unwanted immunological response as it is common in case of similar products isolated from non-human source. Correct. So, both 1 and 2 statements are correct. Next, the first clinical gene therapy was done for the treatment of first clinical gene therapy given to the SID, severe combined immunodeficiency resulting from ADA deficiency. Next question, match the following column 1 and column 2. Conventional method of diagnosis, that is a serum and urine analysis. PCR, it is a amplification of nucleic acid. ELISA, based on antigen antibody interaction. Probe, it is a single stranded DNA or RNA tagged with the radioactive molecule. For this, the first option is correct. The next question, given below are two statements. One is labeled as assertion and other is reason. Assertion, a probe is allowed to hybridize to its complementary DNA in a clone of cells, followed by detection using autoradiography. The clone having the mutated gene will not appear on photographic film. Yes, correct. The probe will not have complementarity with mutated gene. Correct. So both assertion and reason is correct, and reason is a correct explanation for the assertion. So the option 4 is correct. What is the site of production of ADA in the body? The site of production of ADA, that is a lymphocytes. Next, identify the correct set of statements. PCR is being used to detect mutation in genes in suspected cancer patients. Correct. Conventional method of disease diagnosis including serum, urine analysis. Yes, correct. Gene therapy is a collection of methods that allows correction of a gene defect that has been diagnosed in a child or embryo. All of the above. Correct. Yes. The next, transgenic animals and ethical issues. The use of transgenic animals is to test the safety of vaccines, to study human disease such as cystic fibrosis, hemophilia, and rheumatoid arthritis, to study complex factor involved in growth such as insulin like growth factor. Yes, all of the above. Next, which of the following is an incorrect statement? The transgenic pigs are being developed for testing the safety of polio vaccine. Transgenic mice are being developed, not pigs. So the first one is incorrect. 27 documented varieties of basmati rice grown in India. Correct. Patent granted in USA for medicinal properties of neem, turmeric, biopyrus. Yes, of course. 95% of all existing transgenic animals are mice. Correct. Next question. GEAC stands for, that is a Genetic Engineering Approval Committee. The second option. Next, illegal and unlawful development of biomaterials without payment to concerned authorities or organization is called biopiracy. Transgenic models can be used to investigate many human diseases such as cancer, Alzheimer's disease, cystic fibrosis, all of this. Next, which of the following is incorrect with respect to the first transgenic cow, Rosie? It is produced human protein enriched milk, alpha lactalbumin, that is 2.4 grams per liter. Correct. The milk contained human alpha lactalbumin, that is human protein. Correct. It was developed in 1983. This is wrong. 1997. Its milk was a more balanced product for human babies than natural cow's milk. Correct. So the third option is incorrect. Next, assertion reason type. Some ethical standards are required to evaluate morality of all human activities that might help or harm living organisms. Correct. The manipulation of living organisms by human race can go on any further without regulation. This is wrong. The human manipulation of living organisms by human race cannot go any further, not can grow. So assertion is correct, reason is false. So the option second. Next, identify the correct set of statements. Transgenic animals can be specifically designed to allow the study of new genes, how the genes are regulated, and how they are affect normal function of the body and its development. Correct. For chemical safety testing, transgenic animals are exposed to toxic substance and then effects are studied. Yes, correct. The toxicity testing in transgenic animals will allow us to obtain results more time now. This is within a shorter time. The procedures of chemical safety testing in the same as that of toxicity of drug, correct. That is A, B, D are correct. C itself is wrong. The first option is correct. Next, biopiracy means. Biopiracy is the use or exploitation of bioresources without authentic permission. Biopiracy. Next, Indian Parliament has recently cleared the second amendment of the Patent Bill, Indian Patent Bill, which take issues such as patent terms, emergency provision, research and development initiatives to deal with biopiracies. That is Indian Patent Bill. Next, GEAC make decisions. It make a decision regarding validity of GM research, regarding safety of introducing GM organisms for public service, for creating GM food, addressing their safety concern, all of the above. Next question. The milk of transgenic cow Rosie contained the dash and it was nutritionally more balanced product for human babies than natural cow's milk. That is a human alpha lactalbumin, the fourth option. Yes. Next question. Given below are two statements. One is labeled as assertion and the other is reason. Assertion. Transgenic mice are being developed for use in testing the safety of vaccine before they are used on humans. Correct. It could replace the use of monkeys for testing the safety of the vaccine. Yes, correct. But reason is not a correct explanation. So the first option is correct. Next, identify the correct set of statements. Today, transgenic models exist for many human diseases like rheumatoid arthritis. Correct. Animals that have had their DNA manipulated to possess and express an extra or foreign genes are known as transgenic animals. Correct. Indian plants such as basmati rice, turmeric, and neem have been early either patented or attempts have been made to patent them by Western nations for commercial use. Correct. There are an estimated 20,000 varieties of rice in India alone. That is 2 lakh varieties, not 20,000. So option D is incorrect. Correct set A, B, C are correct. The first option is right. Next, given below are two statements. Statement 1. Some nations are developing laws to prevent unauthorized exploitation of their bioresources and traditional knowledge. Next, traditional knowledge related to bioresources can be exploited to develop modern applications and can also be used to save time, effort, expenditure during their commercialization. First and second correct, both are correct statements. So the fourth option is correct. Next, the Indian Parliament has recently cleared the second amendment of Indian Patent Bill that take such issues into considerations. It includes patent terms, emergency provision correct, research, development initiatives. All of the above is correct. Next, identify the correct set of statements. Most of the industrialized nations are financially poor. No, this is most of industrialized nations financially they are rich. The developing and underdeveloped world is poor in biodiversity, traditional knowledge. This is wrong again. Developing and underdeveloped worlds are rich in biodiversity and traditional knowledge. Genetic modification of organisms can have unpredictable results when such organisms are introduced into the ecosystem. Yes, it is possible. The modification or usage of living organisms for public service as a food or medicine source has also created problems with patent granted for the same. Correct. Indian basmati was crossed with semi-dwarf varieties and claimed as an invention or novelty. The patent extends to the functional equivalents, implying that other people selling basmati rice could be restricted by the patent. Here, the C, D, E are the correct statement. First and second, A and B are the wrong. So, the second option is correct. The last question, fill in the blanks. In dash, an American company got a patent right on basmati rice through dash. In 1997, American company got patent rights on basmati through the US Patent and Trademark Office. Third option is correct.